In this video, we learn about light responses in plants. Plants sense and respond to only a specific narrow range of wavelengths in the visible spectrum. The phenomenon of directed response toward light is known as phototropism. This phenomenon was first studied by Charles Darwin and his son Francis. They experimented with coleoptiles, which are protected shoots. The results of their experiments showed that plants respond to light in the presence of the blue wavelength. This is due to the plant's dependency on blue light for photosynthesis. Chlorophyll A and B, which are the prime photosynthetic pigments, absorb strongly in the blue and red spectrum of visible light. Results of several experiments indicate that plants respond by bending toward light only when blue light is present. As we can see in this image, coleoptiles respond to visible light by bending toward it. This response happens specifically in the presence of blue light and not in the presence of any other wavelengths of light that are part of the visible spectrum. Although biologists knew that the blue light receptor must be a pigment, a molecule that absorbs certain wavelengths of light, it took decades to find it. The pigments that absorb blue light are known as phototropins. Researchers found a gene in Arabidopsis called FOT1, which they concluded coded for a blue light receptor. This gene codes for the photo-1 protein, which is activated in the presence of blue light. A particular amino acid that is present within the protein is phosphorylated when the plant is exposed to blue light, which triggers the phototropic response. Scientists discovered the mechanism of activation of the FOT1 protein by growing both wild-type Arabidopsis plants and plants that had a mutation in the FOT1 gene in the presence of blue light. They grew the mutant plants under various conditions. The first involved growing these mutants on their own without any additional treatment. The second condition involved the addition of a mutated FOT1 protein which was unable to be phosphorylated. And the third involved the addition of a functional FOT1 protein that could be activated by phosphorylation. The unaltered wild-type plant would produce a functional copy of the FOT1 protein that can also be activated by phosphorylation. When they measured the amount of phototropic bending in each of these plants, they observed that most of the wild-type plants grew toward the blue light. All of the mutant plants did not show this response. When a mutated form of FOT1 protein was added to these mutant plants, they failed to rescue normal bending. But when a functional FOT1 protein was added to the mutant plants, normal bending was rescued. This response was almost equivalent to the wild-type plants. This indicated to the scientists that phosphorylation of the FOT1 protein triggers phototropic bending in response to blue light. Because a mutated copy of FOT1 protein that was not able to be phosphorylated could not rescue the normal bending response in comparison to a completely functional copy of FOT1 protein that could be phosphorylated, which was able to rescue the bending response in the mutant plants. In addition to the bending response in plants, blue light has an effect on the positioning of the chloroplasts within plant cells. Differences in the intensity of the blue light causes movement of chloroplasts within the cells. In bright light, they move to the sides, shading each other, whereas in low light, they spread out to maximize the absorption of light. In addition to phototropins, there are some other blue light receptors present in plants. For example, the carotenoid pigment zeaxanthin, which controls the opening of the stomata, stem elongation, and flower production. A group of photoreceptors called cryptochromes are also blue light photoreceptors. Cryptochromes are involved in stem growth in shady areas as well as in flower production. After carrying out several additional experiments, the Darwins gathered convincing evidence that blue light is sensed at the tip of a coleoptile and is then transmitted to lower cells. 3 general results were derived from the Darwin experiments. First, removing tips from the coleoptile resulted in the seedlings not bending toward the light. Second, covering the tips resulted in no bending. And third, placing opaque collars below the tip area resulted in bending toward the light. These results led the Darwins to conclude that the blue light sensors are located in the tips of the coleoptile. The Darwins wanted to find out which was the light sensing region and which was the light responding region in the coleoptile. To find out, they set up an experiment where they studied the growth of grass coleoptiles in the presence of light under various conditions. The first served as a control in which the coleoptiles were allowed to grow under natural conditions. The Darwins then removed tips of some of the coleoptiles, or covered the tip of some other coleoptiles, or covered the lower portion of a fourth set of coleoptiles. They observed that the control coleoptiles bent toward light. The second set of coleoptiles did not. The third set did not as well, and the fourth set did. Based on these observations, they concluded that somehow the tips of the coleoptiles are able to sense light and trigger a response further down within the coleoptile to result in the bending of the entire coleoptile toward light. They also hypothesized that the sensing tissue produces a substance that travels down the coleoptile, triggering the bending response. The Darwins hypothesized that the substance, which is produced at the tip of the coleoptile, acts as a signal that is transported to the area of bending. Later experiments in 1913 by Peter Boysen-Jensen and Fritz Wendt supported the hormone hypothesis. Wendt found that decapitated shoot tips gave the phototropic response even when kept in the dark. This observation led him to conclude that a hormone was responsible for the phototropic response and elongation of the shoot tip. Wendt used decapitated coleoptiles for his experiments. When he placed a permeable block of agar on these decapitated coleoptiles and then placed the tips on the agar block, he observed that the coleoptiles bent toward the light. On the other hand, if he placed an impermeable mica block instead of the permeable agar on the decapitated coleoptile and then placed the shoot tip on this mica block, no bending was observed. This led him to conclude that there was a chemical present in the shoot tips that was traveling to the responding cells down the coleoptile, triggering the bending response. In further experiments, he collected this chemical from the shoot tips into agar blocks and placed the agar blocks in an offset position on the decapitated coleoptiles in the dark. He observed that even in the dark, the bending response was triggered in these decapitated coleoptiles. This led him to hypothesize that a hormone which was collected in the agar blocks sent signals down the shaded side of the coleoptile and caused the cells on the side to elongate in response to the hormone, represented by red dots in this image. Wendt named the hormone auxin, meaning to increase, since it triggered the increase or elongation in responding cells. Auxin was the first plant hormone ever discovered. The chemical structure for this hormone was later identified as being that of indole acetic acid, or IAA. IAA is found in minute concentrations ranging from 3 to 500 nanograms per gram of tissue in a typical plant. Experiments by Wendt and another scientist called N.O. Kolodny led to the development of the hypothesis that Phototropism results from an asymmetric distribution of auxin. This was known as the Kolodny-Wendt hypothesis. Their hypothesis makes three predictions. 1. Auxin produced at the tips moves from the light side to the dark side. 2. Auxin is transported down one side of the coleoptile. And 3. This asymmetric distribution causes cells on the shaded side to elongate more than cells on the illuminated side, resulting in bending. This hypothesis was supported by experiments carried out by Winslow Briggs, who found that the asymmetric distribution of auxin results from lateral redistribution. Together, these three scientists and their colleagues explained how auxin leads to asymmetric cell elongation and thus the bending response called phototropism. So how do cells respond to auxin? Auxin-binding proteins are found in the stem and leaf. These auxin-binding proteins are responsible for cell elongation. They have been identified as expansins, proteins that unzip the hydrogen bonds between the cellulose microfibrils, loosening their structure. The signal transduction cascade that follows increases the number of membrane proton pumps, or proton ATPases, in the plasma membrane, triggering cell elongation and leading to phototropism. The acid growth hypothesis for cell elongation is thus termed because the proton pumps drive protons out of the cell against an electrochemical gradient, lowering the pH of the cell wall. Two things have to happen for a plant cell to get larger. 1. The cell wall must expand to create a larger volume. And 2. Water has to enter the cell to increase its volume and generate turgor pressure on the cell wall. When the pH of the cell wall is lowered below 4.5, this activates expansins, which increase cell length by releasing molecular kinks connecting cellulose microfibrils. The electrochemical gradient causes positively charged ions such as potassium as well as sugars to enter the cell. Water follows these solutes by osmosis, causing the turgor of the cell to increase. Most biologists agree that the acid growth hypothesis explains how auxin induces plant cells to elongate. This image illustrates the acid growth hypothesis for cell elongation. The cellulose microfibrils that are present in the cell walls of plants are tightly linked with each other. When a plant cell receives a signal from auxin, proton pumps present within the plasma membrane of the cell use energy from ATP to pump protons. This lowering of the pH outside the cell activates expansins present within the cell wall that unzip hydrogen bonds present between the cellulose microfibrils, resulting in the loosening of the structure, allowing the cell wall to expand. Also, potassium ions enter the cell through potassium ion channels, in addition to other substances like sugars, which lead to the increased uptake of water by the cell, resulting in higher turgor pressure. This pushes the loosened cell wall out, elongating the cell. Now that we've looked at the blue light response in plants, let's discuss the effect of red and far red light on plants. Plants are also sensitive to wavelengths in the red and far red portions of the visible spectrum. Red light drives photosynthesis, whereas far red wavelengths are not absorbed strongly and mostly pass through plant cells. Researchers working with lettuce seeds determined that red and far red light act like an on-off switch for seed germination. Two discoveries were made, one being that the red light promotes germination, and the other being that the far red light inhibits germination. In these experiments, biologists exposed moistened lettuce seeds to alternating flashes of light containing one of two wavelengths, red or far red. After exposure to light, the seeds were held in the dark for several days. As we can see, when the seeds were not exposed to any light, a very low percentage of seeds germinated. On exposure to red light, 98% of the seeds germinated. When this was alternated with far red light, the number of seeds that germinated dropped down to 54%. Almost half of the seeds that germinated when exposed to only red light. As this process was repeated over and over again, we can see that in the presence of red light, most of the seeds germinate, whereas when exposed to far red light, the percentage of seeds that germinate reduces significantly. These experiments showed that red and far red lights served as on and off switches for seed germination. It was later found that the same pigment absorbs both red and far red light by changing shape. One of these shapes absorbs the red light, and a second shape absorbs the far red light. Such switching behavior is known as photoreversibility. The pigment that scientists hypothesized was responsible for responding to red and far red lights was named phytochrome. The shape of phytochrome that absorbs red light is called PR, or phytochrome red. The shape that absorbs far red light is PFR, or phytochrome far red. This image illustrates how a change in the conformation of phytochrome affects germination in lettuce seeds. When the phytochrome receptor present within seed cells is exposed to the red wavelength from sunlight, it changes from the PR conformation to the PFR conformation that is now sensitive to far red light. This results in the germination of the lettuce seeds. When the PFR conformation of phytochrome receives far red light, its shape changes to the PR conformation, and this results in the switching off or inhibition of germination in lettuce seeds. Thus, a conformational change in proteins can tightly regulate processes like germination within plants. This brings us to the end of the section on light responses in plants. After going through this information, I'd like you to be able to respond to the questions listed on this slide. Thank you.